So the next speaker is Toviah Moldwin from Hebrew University of Jerusalem. So he's a PhD student in Idan Segev's group, and his interests are in using machine learning, including decision trees, ANNs, and genetic algorithms to learn biological parameters from experimental data and to use these to simulate biophysically realistic neuron models. So the stage is yours, Toviah.
Okay, y'all see my screen and hear me? Everyone? Okay. Okay, so this is on our work called the Gradient Clusteron. I was on with my student Menachem Kalmanson, and it's in the lab of Idan Segev. Sorry. So practitioners of computational neuroscience and machine learning frequently think in terms of this conceptual mathematical model, the McCulloch and Pitts neuron. In this framework, a neuron is a unit that receives inputs to its synapses. Those are these X's. Each synapse has a weight. Those are the W's. These weighted inputs are summed and then passed through a threshold nonlinearity and gives you an output. You can think of it as a spike or no spike. But it turns out that in biology, neurons are a bit more complicated. So for example, we have this, the NMDA receptor, which is connected to a voltage-dependent channel. So this voltage-dependent creates a synergistic interaction between nearby synapses. So if you activate two synapses next to each other simultaneously, you get this large nonlinear EPSP, which you can see in this orange trace here, which is much larger than what would happen if you would just activate each synapse individually and sum them up, which you see in this blue trace. But if you put the synapses far away from each other and activate them simultaneously, you no longer have this nonlinear phenomenon. An early model by Bartlett-Mell called the clusteron tried to encapsulate this synergistic activity. In this model, synapses are arranged along the dendrite with discrete integer valued locations. The synapses interact with each other in a multiplicative fashion. So the activation of each synapse, which is AI, so let's say the activation of this synapse here, is defined by that weighted input times the sum of the weighted inputs within a particular length of dendrite called the domain, or d sub I. So each of these synapses has this activation, which is the product of that synapse with the sum of the other synapses around it. These activations of each synapse are then summed to produce the output of the whole neuron. These kinds of model neurons are typically used to solve classification tasks, such as looking at an image of a digit and telling us whether it depicts, let's say, the number 0, which we'd call the positive class, or a different number, and then all the other numbers we would call the negative class. So for the clusteron, if it wanted to solve this particular problem, a data set consisting just of examples from the positive class. This is unique to the clusteron. Normally you train on both positive and negative, but for the clusteron, you present images just from the positive class. You calculate the average synaptic activation on the entire data set, and synapses which have less than the average activation are randomly shuffled with each other. And what that does is it sort of increases the probability that correlated synapses within this positive class of images will sit next to each other on the dendrite, thus maximizing this synergistic coactivation. This model was subsequently expanded to the two-layer model of Yoda Parazi and also Bartlett-Mell, and they traded the multiplicative nonlinearity for a sigmoidal nonlinearity. But for now, we'll focus on this single branch neuron. So together with my student Menachem Kalmanson, we took this single branch clusteron in a new direction. We wanted to improve the clusteron's classification ability by creating a model with an analytically tractable gradient descent algorithm. And we call this model the G-clusteron. So we made two major modifications to the original clusteron. The first modification is that we swapped the discrete locations from the original clusteron with continuous valued locations. So each synapse is now associated with a location, which we call L, instead of an integer valued index. And so for, let's say, synapse 6, so location 6, let's say it's 0. But if you take this synapse and move it over here, it will still be called location 6, but location 6 might equal 3.2. The second modification is that instead of each synapse multiplying the other synapses within a fixed range on the dendrite, now the interaction is based on this distance-dependent function. So if you look at this bottom figure here and look at synapse 1, so it interacts with itself with a factor of 1. That's the maximum. It interacts with synapse 2 with a factor of 0.5 because it's kind of far away, and it interacts with synapse 1 interacts with synapse 3 with 0.2 because synapse 3 is even further away from synapse 1. And you can just see that in this equation here. It's the same as the original clusteron, but instead of multiplying everything in your domain over here, instead you're applying this distance-dependent function Fij, which depends on the distance between every synapse. Okay, so we want to make this thing learn, and we want to have a gradient descent algorithm. So we derived the following plasticity rule for the locations. What this rule does is it moves the locations of each synapse. Basically, the idea is that synapses attract or repel each other, and we sum over all of these attractive or repulsive forces for each synapse to calculate where it should move. These forces depend on the target value, so the positive or negative class, the weighted inputs to the synapses, and the distance between synapses. So look at C. So in the positive pattern, that's the one that the neuron is supposed to produce a large output for, or a spike. So synapses with the same sign, so that means W times the weight times the input. So if this has a positive weight times input and that also has a positive weighted input, they're attracted to each other. The same thing if they're both negative, they're attracted to each other. But if one synapse has a positive weighted input and the other synapse has a negative weighted input, then they repel each other. And for a negative pattern, so when you want the neuron to produce a smaller output, so it's reversed. So opposite sign inputs repel and same sign inputs attract. And the reason for this is pretty simple, because this is a multiplicative rule. So a negative times a negative is a positive, and a positive times a positive is a positive. So if you want to increase the activation, you want to put positives together and negatives together. And if you want to decrease the activation, you do the opposite. So that's the weighted input dependence. There is a dependence on also the magnitude of the weighted input, which is straightforward. It's just the magnitude of the attraction repulsion depends on the magnitude of the weighted input. And then you also have a distance dependent aspect to it. So if synapses are close together, they will not have a strong force on each other. If they're intermediate distance, the force is larger. And if they're very far, again, the attraction between them drops off. So in short, if the clusteron algorithm is sort of like randomly swapping seats in a movie theater, because you have discrete seats, discrete locations, and the algorithm just randomly swaps them. So the G-clusteron is more like sliding on a bench towards or away from people who you like or don't like. Okay, so we're now going to use this on a practical classification task. The idea here is that we're going to classify MNIST, and what we want to happen is that pixels which are correlated in the positive class. So here we're interested in classifying the digit 2 versus everything else. So we want the black pixels in the 2 to appear next, or in all of we want the black pixels in the 2 to appear next to each other on the dendrite, and we want the white pixels in the 2 to appear next to each other on the dendrite. But if we look at other digits and we look at those same locations in the digits, it won't be that way, right? So here, the green and blue pixels, the green and blue circled pixels show a black input and a white input, and when they're next to each other on the dendrite, they'll interfere with each other and have a smaller activation. So that's basically the idea. Put correlated inputs in the positive class next to each other on the dendrite, and for the negative class, correlated inputs should be far away from each other. And just to get to the accuracy on the all-versus-all classification task, so we're trying to now, instead of just doing is this a 2, is this not a 2, we now want to classify the entire MNIST data set. So I'll give this classifier digits from 0 to 9, and it will tell me which digit it is. So in order to do that, typically there are 2 ways to do an all-versus-all classification task. Either you can train each classifier for each digit independently, or what you can do is that you can send them to a softmax function. And the softmax, essentially what it does is it normalizes the output of each classifier with the output of all the other classifiers. So it sort of allows the classifiers for each digit to communicate with each other, and this normally gives you better classification accuracy. So importantly, the original clusteron actually cannot use this softmax function because the original clusteron doesn't actually have an error term that you can propagate out of the softmax function. So that's really a strong benefit of the G-clusteron over the clusteron. And so we show that if you train the clusteron on this all-versus-all MNIST task using this one versus rest procedure where you don't have a softmax, so you get around 60-something%. And the same thing with the G-clusteron. But if you add the softmax layer to the G-clusteron, so you just allow the classifiers to communicate with each other, you get around 86% accuracy, which is basically the same as you get with logistic regression, which you can think of essentially as a neural network without any hidden units. And remember, this G-clusteron, so far, we have not modified any synaptic weights. So, right, so we've managed to get within 5% of logistic regression in terms of accuracy, while only modifying synaptic locations and not modifying synaptic weights. But this, the model that we originally showed, if you notice, it actually did have weights in it. So what are we going to do with those weights? Before, we had just set them all to 1 and pretended that they didn't exist. But we actually derived a learning rule for the weights as well, which is much simpler. It's just the error times the activation divided by the weight. So now we can modify the weights. It turns out I didn't show it here, but on MNIST, it doesn't make such a big difference if you have just the weight rule or just the location rule or both. But we show that actually, if you have you can solve the exclusive OR problem with the G-clusteron if and only if you are able to modify both the synaptic weights and location. So I won't get into this in too much detail, but this is sort of the solution space for the G-clusteron in terms of its weights, and F12 is the distance dependent factor, but you can think of it as the location. So to solve exclusive OR, just to remind you, right, it's a binary function. You put in 2 bits. If the bits are different, right, one's a 0 and the other is a 1, it returns a 1. If they're the same, they're both 0s or both 1s, it returns a 0. So the way that you solve this on the dendrite is that you have 2 synapses, and each synapse has a weight, and the synapses also have a certain distance from each other. And we show both analytically and numerically that if you want the G-clusteron to be able to learn to solve the exclusive OR problem from arbitrary initial conditions, you basically have to set both the weights and the locations correctly. So if you have, let's say, 2 of the same sign, so 2, let's say, excitatory weights, 2 positive weights, and they're next to each other on the dendrite, that doesn't work. If they're opposite weights, +1 or -1, but they're far away, that also doesn't work. But if they're opposite weights and also next to each other, then that does work. And we show that numerically too. So we just we run the G-clusteron on the exclusive OR task for 1,000 iterations, and then we just show numerically that if you have both in the weight and the location rule, you can basically learn it from arbitrary initial conditions. But if you have just the weight rule or just the location rule, then you can only basically solve the problem if it was initialized in a sort of fortuitous manner. So to summarize, the G-clusteron is a modification of Mel's 1991 clusteron. The differences are that the locations are continuous and the interaction between the synapses are based on a smooth distance-dependent function. It learns with gradient descent. If you have softmax, then the G-clusteron is much better for multi-class classification than the original clusteron. It's about as good as logistic regression, again, if you use the softmax. And if you have both the weight and location update rules, then it can learn to solve the exclusive OR task from arbitrary initial conditions. I would like to thank my whole lab, in particular Malcolm Kalmanson, who is really, he did most of the coding for this. And also Ayal Gal from our lab, and Itam Orlando from Chaim Sampolinsky's lab, and Nidan Segev, my professor. I do not have a bioarchive version of this paper out yet, but it should be out soon. So if you follow me on Twitter, if you follow the lab's Twitter, I hope to have a version of it out within the next couple of weeks. So thank you.
It's great. Thank you for the wonderful talk, Toviah. And Do we have any questions? We do. So Brendan Bicknell asks, are the weights sign constrained, that is explicit E&I synapses? And have you tried multiple dendrites?
Okay, so the answer to that is, so first of all, the weights are not sign constrained, but initially when we learn MNIST just with the locations, the weights are all fixed at 1. The inputs are different. So actually, in order for it to learn well, you have to normalize the input. We have to basically subtract the mean. So half of the inputs will be negative. But no, the weights in this model are not sign constrained. And the second question again was, multiple dendrites. So we've thought about it. We haven't implemented that. That's one kind of interesting direction. Another interesting direction could be high dimensions, because we did this in biology. Of course, dendrites are basically one-dimensional, but the algorithm could in principle work for a high-dimensional dendrite, so to speak, as well, because there's nothing forcing it to be in one dimension. But yeah, it's an interesting direction.
That's great. And